This lecture is going to focus on the rise of animal diversity, and it's going to cover chapters 27 through 29. There are a lot of pages in those chapters, so use this as your guide as you're going through. Make sure you're writing down your notes. Now, animals originated more than 700 million years ago. Our earliest fossils, where our biochemical analysis is showing somewhere around 770 million years. So animals likely evolved from single-celled eukaryotes that are similar to protists or the choanoflagellates that we find today in our sponges. They're members of Opistheconta, which means behind pole. That's referring to the single flagella at one end of the cell that allows for locomotion and generation of flow. Now, 1.3 million animal species currently have been named, but there are likely to be around 8 million animal species. As mentioned, fossil biochemical evidence and the molecular analysis working our way in reverse gives our last common ancestor to about 770 million years ago. That is a long time. That was actually before plants. So animals actually arose before plants did. So the earliest animal fossils are from the Ediacaran biota. So that dates around 560 million years ago. They're still soft-bodied. They haven't started the arms race just yet. They're working on it, but we're going to talk about that arms race as we continue throughout the rest of this lecture. So Dickinsonia is here, Cambriella is here. That's a type of mollusk. So we have these fossils, and we can find those in the Ediacaran Mountains of Australia. Now, sponges and cnidarians are early diverging groups of animals. Sponges, you may be familiar with those from, the bath or whatever. Sponges are aggregates of cells. They don't have any true tissues, so they're actually in their own classification. Cnidarians are jellyfish. They do have true tissues, but they only have 2 of them. Later on, we'll talk about the third type of tissue. So we start seeing, no cell walls, unlike plants and fungus. We start seeing an extracellular matrix being generated so they can start working together. They can, anchor themselves together. They can start supplying different types of tissues, if you will. But with that, we start seeing our true tissues arise. So sponges, cnidarians, and all of the other animal groups start to diverge from one another once this process begins. So let's look at the simplest first. Sponges have been around forever, really, 700 million years or so. So they are, suspension feeders, which means they start working together to generate a flow so that the water is pulled through their cells and it gets stuck inside that extracellular matrix. So as the water passes through that body, then the suspended food particles, those get captured. Those suspended food particles can then be shared. Now, sponges lack true tissues, which means they can't use the tissue itself to anchor it. They can't use the tissue itself to move those nutrients, but there is a shared unit here. But watch what happens as the food gets pulled in through these little pores. The food gets trapped, it gets caught by these little collar cells, which can then share all of the nutrients to the rest of those cells. So the cells have learned to work together. Some of them are amoebocytes, and they'll work as kind of a glue, if you will. And the food particles are passed from a choanoflagellate to amoebocytes. See how the food is moving? The way they generate that flow to pull that through is to create a flow using the flagellum. And remember, these are opisthecons, which means they've got a flagella on one end. So as they generate that flow, the water moves outward. That creates, if you will, a negative pressure. That flow will then pull more water in through these little pores on the side. They've learned to work together. So the choanoflagellates are those flagellated collar cells. They resemble the choanoflagellates. They generate that water flow, and the water flow is going to allow the ingestion of that suspended food, any of the nutrients that are floating around in the water. The amoebocytes, those are these mobile cells that will then play a role in digestion structure. They allow for anchoring, kind of like a glue, if you will. And then molecular morphological similarities between sponges and choanoflagellates suggest that the animals evolved from a choanoflagellate-like ancestor. And I'll show you one of those later. Now, cnidarians, these are members of the phylum Cnidaria. They do have true tissues. They have 2 of them. Unlike sponges, if you tear a cnidarian, the animal dies. Sponges, if you tear a sponge, you just end up with 2 sponges. That's because those true tissues are actually working together to maintain the structural integrity of the organism. Now, Cnidarians are one of the oldest groups of animals. They date back to about 680 million years. We do have some of their fossils. They're rare, but we do have some of them dating back to that time. Cnidarians have diversified into a very wide range of sessile, which means they're anchored to the bottom, and motile forms like medusae. We also include hydrozoans, jellies or jellyfish, and sea anemones. They look a bit like this. So we have our hydrozoans. Generally, hydrozoans are roughly, they're anchored, they're relatively small, but you're going to see them in, large quantities kind of gathered together. They're not all like that. Some of them are individuals, but we see this quite often. Scyphozoans, such as the jellyfish here, those have a sessile polyp stage and a motile medusa stage. So they have a reproductive stage that's actually going to get up and move around. And then the anthozoans, those are our sea anemones. And in case you've ever wondered, can sea anemones as animals get up and move around? The answer is yes, they can. They can actually swim. They're not very good at it, but they can avoid predators like the starfish. So the basic body plan of Cnidarians is a sac. That's really it. It's got a central digestive compartment called a gastrovascular cavity. The tentacles try to pull the food close to that gastrovascular cavity so that you allow the nutrients to, just by simple diffusion, move across from the decaying animal into the jellyfish. So that single opening functions as a mouth and an anus. So you're going to have food brought in, you're going to have waste products sent out. Cnidarians are carnivores. They use their tentacles to capture the prey, and they have a non-centralized nerve net. So it's kind of like a primitive brain. But these types of cells actually arose independently of the same types of neurons that we use. Now, there was a period of time known as the Cambrian Explosion. This was after the Precambrian. The Precambrian did start to see, life, of course, arise. But during the Cambrian Explosion, this is where we start seeing the fossil appearance of many of the major groups that we still see today. So this explosion was a rapid diversification rather than an actual explosion. Rapid diversification of these life forms means that they started an arms race. An arms race. Who could outperform the other? Could they become, more structured? Could they become more bony on the outside so they would avoid predation? Could they become faster? Could they see better? And so on. So they started arguing back and forth genetically on who was going to be the most supreme, if you will. So the strata that was formed during the Cambrian start showing the oldest fossils for half of all of the extant animal phyla, which means half of the animals that are alive now found their fossils showing up right here in the Cambrian Explosion. So, for example, we see the no true tissues during the early portion of the Proterozoic. We see the Cnidarians start to branch off during the Ediacaran. And then we start seeing later on during the Cambrian Explosion, the others start to branch off. Okay. So somewhere about 540 million years ago or so, we start seeing half of the animals that we currently have branch out. So fossils from the Cambrian period include the very first hard mineralized skeletons, those likely developed for defense. We also start seeing, animals become bilaterans. Bilaterans mean that they have a left and a right. They're going to be bilaterally symmetric. They have a complete digestive tract, which means they've got a mouth in the front, if you will, and an anus in the back. So we've got a front and a back, a left and a right, and a top and a bottom. That allows for animals to move around in the water very efficiently. So the very first hunting predators start diverging in this period of time. Same thing with the eyes. We start seeing the eyes start to develop so that we have this predator-prey relationship. And they look a bit like this. Now, some of these animals may look very unfamiliar to you, and that's okay. Things like the hallucinogenia. Hallucinogenia, this little animal here, we originally thought was upside down, but we found out later on that we were wrong, and that's okay. We realized that the animal had a front and a back, left and right, up and down. We just reoriented ourselves to the animal itself, at least the fossils. Others had a mouth in the front, tail at the end, left and right. top and bottom, slightly different from one another, but left and right allowed them to be bilaterally symmetric so that they could move efficiently through the water. There are several hypotheses that lead up to the, Cambrian explosion. Why did it happen? And why did the Ediacaran biota kind of die off? So new predator-prey relationships, they were fighting back and forth with each other. Those that could keep up did. Those that didn't went extinct. Remember that 99% of all animals that have ever existed are now extinct. We also saw a rise in atmospheric oxygen, and we saw the evolution of the Hox genes. Now, these Hox genes, these are multiple types of genes, but that allows for that head-tail axis, or otherwise known as bilateral symmetry. Things sped up. We were becoming more efficient. So the molecular clock estimates bilaterians to about 100 million years earlier than the oldest fossils. So that, would have been 560 million years ago or so. Excuse me. The appearance of larger, well-defined eukaryotes. So we've got 635 to 542 million years ago, starts to show that bilaterians, those predators may have originated by that time. It doesn't seem like much, but when you have 100 million years of predator-prey relationship, something is going to give, honestly. That something that gives allows those organisms to develop the very best way that they can survive in their niche. If they can survive in one area, whereas something else cannot, they're going to then thrive. So you start seeing this thrive kind of genetic debt, if you will. If they can thrive, they do. And if they don't, that genetic debt leads to them becoming extinct. They can't keep up. They can't outperform others. And if they can't outperform others, something else will then displace them. So we start seeing these soft-bodied, organisms turn into, hard spine-covered organisms. The reason why they started becoming bony or hard was for predation. It was for predators. Keep the predators away, and you don't have to worry about it. You're not an easy food group. So animals in the early Cambrian oceans were very diverse in morphology, as we showed just a minute ago. They had a different way of life. They found different types of niches that had never been occupied before, and lots of taxonomic affiliations. So you'll see more taxonomy in just a second. So zoologists recognize about 36 animal phyla. Phylogenies now combine molecular data that we've found from multiple sources with the morphological data as well. So you start looking at how are they shaped? Why are they shaped the way they are? And then we also look at the molecular data such as RNA and DNA. We find that the similarities between those two then allows a better type of classification. So they look a bit like this. So we start off with our metazoans. The metazoans, meta meaning middle, so middle age zoa are animals. We've got our eumetazoa, the true middle-aged zoa or animals. So porphyrians branched off, then the ctenophorans and cnidarians. Ctenophorans are types of sea jellies. They're not jellyfish, but they're types of sea jellies. They're beautiful to look at if you ever get a chance to see any of their photophores, because they can change color. It's brilliant. They actually generate their own light. They branch off from cnidarians, true jellies, as well as the cyphozoa and so on. And then we start seeing bilateria. We are going to have two different types. We have the deuterostomes and the protostomes. Protostomes, their mouth forms first. Deuterostomes, their mouth forms second. Deuterostomes include hemichordata, the echinodermata, which are starfish, and chordates. Those are things like fish and humans. So we're actually more closely related to starfish than we are to things like crayfish. Now, with protostomes, you're going to see most of the animals in this course that we talk about over here, okay, between the ecdysozoa and the lophotrochozoa. So the animal body plans, I actually uploaded an article about these body plans, why we only have 5 left. So you can put that, I've posted it in the Blackboard shell. You can see that. Please look through it. Take your time. I'm not going to test you over it, of course, but it's a good read, and it kind of explains how did we get where we are now. Okay. So these body plans are sets of integrated morphological and developmental traits. So what they try to do is they're showing the comparisons between certain key animal features, what shows up and when and why. Why were they successful or why were they not successful? So the three important aspects for animal body plan: symmetry, tissues, and body cavities. We've talked about symmetry. We've talked about tissues. We're going to get further into both of those, but we haven't talked about body cavities just yet. We will. Now, symmetry, the animal can be categorized as having symmetry or not. So they can be symmetrical or asymmetrical. Those that are symmetrical, are they symmetrical all the way around or just the left and right? If they're symmetrical all the way around, we call that radial symmetry. There's no front or back, left or right. Starfish are this way. Two-sided symmetry, or left and right, is known as bilateral symmetry. Here's an image of those. So we've got something like a sea anemone. Those are radially symmetric. They're the same regardless of how you cut them. Kind of like a pot. So they've got a top and a bottom, but they don't have a left or right, front or back. Whereas those that are bilateral, they have a left and a right, a front and a back, a top and a bottom. Now, because of that, we have to orient ourselves when we're looking at an organism. The way that we orient that is by our anatomical terminology, such as the dorsal side or the back portion of the animal, the ventral side or the belly side of the animal. The cephalic or anteropostural axis allows for an anterior end. The anterior end is where the head is, and a posterior end or caudal end. That's where the tail is. So we have these specialized terms to allow us to orient ourselves as to how we're, describing an organism. Bilaterally symmetrical animals have a dorsal side, a ventral side, right and left, anterior and posterior. We use these terms transverse, sagittal, and coronal. Transverse means you're going to separate the top portion, or excuse me, the dorsal portion from the ventral portion. Okay? Transverse goes for dorsal to ventral in this example. Left and right is going to divide sagittally. So you're actually going to divide straight down the middle of the body, start from the head, work your way down to the tail, and you're going to separate that left and right. Whereas the anterior and posterior, or the front and back ends, are divided coronally. In human beings, a coronal suture runs across. If you would place a crown on your head, where the front portion of that crown would sit on top of your head, that line is the coronal suture, and it's going to divide the anterior portion from the posterior portion. Now, in most animals that are bilaterally symmetric, the sensory equipment, the eyes, the ears, and so on, those are going to be involved in the anterior end because that's how they're going to interact with their world. The posterior end is going to be used for propulsion. Now, radially symmetrical animals, those are generally sessile, or at least temporarily sessile. They are attached to a substrate, which means they're going to grab onto the rocks. Or they're planktonic. They kind of drift or they swim just a little bit. For example, jellyfish. Bilateral animals move actively. That means they're going to swim. They are actively going to put energy in to make sure they're moving as quickly or as efficiently as possible. The central nervous system allows that coordinated movement. Now, tissues, we start with no tissues, like a sponge, and then we get into two tissues. Well, what about those that were just one? They went extinct and didn't work as well. So those tissues are collections of these specialized cells that allow it to work as a functional unit. So we will talk about functional units a little bit later on in this course when we get into specialized systems. But these are going to be isolated from other tissues, such as muscle tissue is different than skin. So during the development, as the embryo is developing, these germ layers start to give rise to these tissues. The tissues can then specialize into organs. Those organs have specialized functions. So we start seeing this division of labor. If you do this, I'll do that. So we can see these tissues arise in the animal embryo. Now we have ectoderm and endoderm that we're going to start with. We have one more here, mesoderm. I'll come to that in a second. Ectoderm, that's going to be the outer germ layer. It gives rise to the coverings like our skin it also gives rise to the central nervous system. So I've shortened it here to nervous system and skin, or integument. The endoderm is the innermost germ layer, and it's going to give rise to the digestive tract, the organs that are involved with that, such as the liver. It's also going to involve the lungs. So if you will, put it in quotations because it's kind of hazy, just everything else that isn't muscle or bone. Okay, so mesoderm. This is not found in every type of animal. Okay, like cnidarians. They don't have any mesoderm. They do, on the other hand, have ectoderm and endoderm. Does that mean they have lungs? No, it doesn't. They don't have lungs. But in those animals that do have lungs, the endoderm is what gave rise to that. So mesoderm, muscle. So it gives rise to the muscle, the cartilage, and the bone. Basically, the internal structure, the internal skeleton. Ectoderm, nervous and skin. Mesoderm, muscle. Everything else, endoderm. If you're going to take a stab in the dark, like you really can't figure it out, you're not entirely sure, best bet, endoderm. Okay. Now, mesoderm, again, that's going to fill the space between the ectoderm and the endoderm. It's going to give rise to the muscles and most of the other organs, specifically the bone, the muscle, and the cartilage. Mesoderm, muscle. Now, some of those will actually have some spaces as well. These are known as body cavities. Can you see a little space right there? Those body cavities allow the digestive tract to be separated from the outside layer. That allows the digestive tract to operate independently while the rest of the body is doing what it needs to do, such as looking for food. So those body cavities separate the internal structure from the more superficial structure. Otherwise, every time you would eat, your whole body would just get as solid as possible until the food then passed out. So we see an increase in mobility and an increase in motility during all of this. Now, with the development of the mouth, we can have the protostome or the deuterostome. Protostome, the mouth forms first. The deuterostome, the anus forms first. We're also going to find that for a protostome, they diverge or they start to develop through spiral cleavage. Deuterostomes use radial cleavage, and I'll show you a picture of that here. So for a protostome, here's the blastula. That's the original cells as they're starting to form the embryo. The blastopore becomes the mouth first, and we see the mesoderm start to arise. They will start to separate out, and they will create the body cavities here. Call it a coelom. So as the mesoderm starts to split, it creates a mouth on one end, an anus on the other, as it starts to develop, with the body space in between, the body cavity. Deuterostomes, on the other hand, start off as a blastula. Their blastopore is the anus. We then start to see a split with the mesoderm filling in that body cavity space here, creating a coelom. The anus then continues to develop into the mouth. You notice that there is a mouth on one end, anus on the other, mouth on one end, anus on the other, still have a coelom, but they came to the conclusion through two different processes. Okay. Those that are deuterostomes, those are our echinoderms and chordates. Everything else, protostome. Now, those body cavities, most bilaterians, they're going to have a body cavity called a coelom. So it can be fluid-filled or air-filled. Either way is fine. But what it does is it separates the digestive tract and the outer body wall. So it cushions the suspended organs. It acts like a hydrostatic skeleton if necessary. And many of you have experienced this. When you go to pick up something heavy, you kind of bear down a little bit. That creates a hydrostatic skeleton. And then it also enables the internal organs to move independently of the body wall. And that's probably the most important. Now, with animal phylogeny, all animals share a common ancestor. Sponges are the basal animals. Eumetazoa is a clade of animals with true tissues. Eu means true. Most animal phyla belong in the clade Bilateria. That does include some of those that, as an adult, are radial asymmetric, like the starfish. And most animals are invertebrates. They don't have a backbone. Chordata is the only phylum that includes vertebrates. Everything else doesn't. So if you're really going to study things that are in the ocean, most of the organisms that you'll study will most likely be invertebrate. So bilaterian radiation number 1, we start seeing diverse invertebrates. So bilaterians start to diverge into the 3 major clades: Lophotrochozoa, Ecdysozoa, and Deuterostomia. Bilaterian invertebrates account for 95% of the known animal species. So if you're going to take a stab in the dark at what that weird animal name is, it's most likely going to be an invertebrate. They are morphologically diverse. They occupy every inhabitant on the planet. And the vast majority of invertebrate species belong to Lophotrochozoa and Ecdysozoa. So for example, these little rotifers here, Ectoprox, and a fireworm. Those are all Lophotrochozoas. We've got a roundworm, and our arachnids here, we've got Arthropoda. Those are both Ecdysozoa. And then the Deuterostomia, starfish, and chordates. Lophotrochozoa are the most morphologically and taxonomically diverse of all of the bilaterians. So they provide most of the invertebrate radiation. The phylum Mollusca, that's going to involve somewhere around 100,000 species or so. And the body of a mollusk has 3 main parts. It has a muscular foot that's used for movement, a visceral mass that contains the internal organs, and a mantle. The mantle can secrete a shell if it's present to do so. Things like octopus don't do that. But it drapes over the visceral mass, covers up all of the internal organs to try to keep them safe. But that muscular foot is actually how we name them. It's how do they interact with their world? Do they interact with their world through their stomach, or do they interact with their world through their head, like a cephalopod, as compared to something like a gastropod? So let's look at a gastropod. So we've got our muscular foot down here. You see the little foot, all of that maintains the nerve cords. That's going to allow the animal to interact with its world. It has a mouth with a radula. It's kind of a scraping, kind of a tongue that's got teeth on it, but it's no teeth, and it doesn't really have a jaw. But it scrapes, like it's going to scrape algae off the rock. Once the food is scraped off, it then enters, the stomach. It'll go through the digestive system and then back out. So all of this area right here, known as the visceral mass, contains the internal organs. The foot is how it interacts with its world. And there are lots of different types as well. And then surrounding or draping over the visceral mass is the mantle. If the mantle can secrete a shell, it does. Okay. Things like abalone and snails will create a shell. Okay. Now, there are some that don't. Nudibranchs are some of those that don't. They don't secrete a shell at all. But those are sea slugs. Bivalves mean you've got 2 shells, one on one side and one on the other. The little black dots are eyes. They can see you. But those bivalves have a shell on one side and a shell on the other. So they can secrete it. Cephalopods, cephalo meaning head and pod meaning foot, they're head-footed animals. They interact with their world through their head. Their visceral mass is back here, containing their internal organs. Their eyes, their tentacles, those are what they use to interact with their world, feel and move around and capture prey and so on. But in this case, they don't secrete a shell. The mantle doesn't do that. Now, arthropods, there are about 1 billion on this planet right now. There are about 1 million arthropod species. So 1.8 million known animal species, 1 million of those are arthropods. They are found in nearly every habitat in the biosphere. 10 kilometers up, 10 kilometers down. Anywhere you can go on the planet, you're going to find arthropods. So the diversity and the success of this group is attributed to their body plan. It's a brilliant body plan. They have a segmented body, they have a hard exoskeleton, and they have jointed appendages. The segmented body allows them to have a shared or a division of labor. So the head region, the thorax, and the abdomen will all do different things. A hard exoskeleton, allowing for them to interact with their world without having to worry about things like predation. And then the jointed appendages allow them to move. So it starts showing up about 535 million years ago or so. And early arthropods show very little variation from segment to segment. They roughly look the same, such as this trilobite. The segment from here to here to here to here are roughly all the same. Tapering a little, but that's about it. So they had a head region, That allowed them to migrate forward. And then all the rest of the segments kind of look the same. So arthropod evolution is characterized by a decreasing number of body segments. So we no longer have, hundreds of these segments. We only have 3. We still have some of these arthropods around that have multiples, like millipedes and centipedes. But we start seeing a regulation of the existing Hox genes. You use those Hox genes that allowed for head region, tail region, left and right, top and bottom. You alter those Hox genes just a little bit, and you get a new function for the animal, such as turning a walking leg into a feeding appendage, such as we see with things like spiders and scorpions. So once we see the origin of these Hox genes, we start seeing that there is a divergence. Okay? So things like velvet worm. So red indicates the region, in this case, where the genes were expressed as compared to the others. If those genes are present, you'll get one type of body segment. If they're not present or not being expressed, they won't. So you'll get these completely different types of development. In case you wonder how important those Hox genes are. Now, vertebrates, these have been the ocean's dominant predator for more than 400 million years. Now, when did animals arise? 770 million years ago. So for roughly 370 million years, or half that time, if you will, the invertebrates dominated. But once the chordates started to arise, they took over, and they did a very good job of it. So vertebrates are named for vertebrae. That's a series of bones that make up their backbone, as well as their notochord, and I'll talk about notochords later. But one of the earliest close relatives of the vertebrates evolved in the ocean during the Cambrian period. So we've been around a long time. Head region, tail region, and vertebrae. Vertebrates are members of the phylum Chordata. Chordates are bilaterian animals. They belong in the animal clade Deuterostomia. I mentioned that earlier. So start off in the water. Later on, we'll get out on land. So all chordates have the 4 derived characters. A notochord, which is a flexible rod that provides support. A dorsal hollow nerve cord. We actually have a little tube, a little hole that goes down the spinal cord. It's called the central canal. That's then going to develop into the brain and spinal cord. We have pharyngeal slits or pharyngeal clefts. Those function in suspension feeding, such as gills. Or later on, they can develop into parts of the head, and then have a muscular post-anal tail. Some maintain that tail, others it doesn't really grow. Human beings don't really have a tail, but we do have a tailbone. Lancelets are our basal groups of extant blade-like animals that most closely resemble the idealized chordate. I'll show you a picture of that in just a second. Tunicates, on the other hand, they are an early diverging chordate group. They do have the notochord to begin with. They kind of look a bit like a tadpole or a polywog once they hatch, but then they land and they go through this kind of metamorphosis and turn into kind of an underwater heart. It looks like an underwater heart. I would love to be able to describe that better. I don't have the words to do so, but when I show you one, you'll understand. The ancestral chordate may have looked similar to a lancelet, and those look like this. So we have our dorsal hollow nerve cord, a cephalic region. Even though they don't have a true brain, they do have a cephalic region. Apologize. So the food comes in one end, goes through a complete digestive system, and then exits the anus. Notice this anus then has a post-anal tail. They have muscle segments. So we're starting to get the 3 different types of tissues, and we've got notochord. That notochord allows those muscles to attach so that then can use those attachment points to create swimming motion. They are motile. They get up and move around. Half the time, however, they work as filter feeders. So they'll pull in the nutrients from the water while stabbing themselves down into the soil. And if they ever get disturbed, they just get up and swim around until they can find some other place. Now, here's the tunicate that I mentioned earlier, kind of the underwater heart. They have this system where they pull water in, they filter it using these pharyngeal clefts and pharyngeal slits, then becomes a stomach, and then it will then send that out. So we have an in siphon and an ex siphon. Those are very primitive. Now, in addition to the features of all chordates, early vertebrates also had a backbone, a skull, and a very well-developed head with a brain on the inside. The brain allowed the sensory organs to interact with the world, eyes, ears, and so on. Fossils representing that transition start showing up in the Cambrian Explosion. That gives us the rise of the vertebrates. So somewhere between 770 million years ago and 400 million years ago, they just kind of averaged that at about 500 million years. Early vertebrates were more efficient at capturing food in the water and evading predators than their ancestors were, which means they were just going to dominate the oceans. So the earliest vertebrates in the fossil record lacked jaws. Oh, they didn't have jaws. Well, how did they bite things? Cool thing. They just used suction. They'd grab on to pull on, and it would stab the teeth into that tissue. So two extant lineages of jawless vertebrates are still with us. We call them the cyclostomes. Cyclo meaning to go around. So the hagfish and the lampreys. I'll show you those in just a bit. Now, today we have the jawed vertebrates, or otherwise known as the gnathostomes. Gnatho means to chew. Those outnumber the jawless vertebrates, which is why you may not have ever heard of lampreys or hagfish. So early gnathostomes start seeing their adaptations really become effective. Those adaptations for predation, that's the arms race again. You start seeing paired fins, one on one side, one on the other. You start seeing tails that are built for efficient swimming. They're not just kind of flopping around in the water. They are able to hold their head still while controlling that tail just enough to increase their efficiency in moving through that water. Then we start to see the development of the jaws. The jaws then grasp onto the prey or take out chunks from the prey. You don't necessarily have to kill it or eat it all in one bite. You can take out chunks, but they can't chew it yet. So gnathostomes diverged from three surviving lineages. Now we have the chondrichthyans. Those are our cartilaginous, I apologize, I said from, and I should have said to. The gnathostomes diverged into three surviving lineages. The chondrichthyes, those are cartilaginous fish, sharks, rays, and skates. We also see them start to diverge into the Rayfend fish and into the lobed fin fish. So the actinopterygi and the sarcopterygi. Humans and other terrestrial animals are included in the lobed fin fish. We're actually a land-born lobed fin fish. All right. So we start seeing the jaws develop, and then we get lungs or lung derivatives. So we see the Rayfend fish, the actinopterygi that I just mentioned. Those are the Rayfend fish, actinopterygi. And then the others where we start seeing the lobed fin fish, those are the sarcopterygi. Okay. But I've got examples of each one of these. Really prime example here, this fish is actinopterygi. Can you see that those are all Rayfend, which means it's kind of diverge and go in each direction? Actinista, that's a coelacanth here. Okay. The coelacanth, that's a sarcopterygi. Okay. Alrighty, Dunkleosteus, one of my favorite. They actually had two skeletons. They had a skeleton that developed inside their skin, and they had an internal skeleton as well. Dunkleosteus, this guy was a shark eater. The skeleton on the outside was actually allowing for these kind of massive jaw structures to sharpen themselves every time they would chomp down or bite. Now, the front end of this animal has been found. The back end of this animal has not. So what does that tell you about what the back end of the animal is built of? If the back end of the animal doesn't fossilize, what's it made of? Is it something made out of hard tissue or soft tissue? Well, if it's made out of something that doesn't fossilize, how do we know what it looked like? We take models of animals that look similar to this now and just scale it back up. That's how we know what they look like. Now, do we know exactly what it looked like? No. Does that mean that we don't have a really good guess of what it looks like? You know, I love this fossil. There's only two full Dunkleosteus fossils on the planet. Now, the chondrichthyans, those are sharks, rays, and skates. So all of their relatives. Skeletons of the chondrichthyans are composed primarily of cartilage, which means they also don't fossilize as soft tissue. Now, that doesn't mean they haven't fossilized. It just means most of them don't. Well, if most of them don't fossilize, those that do then allow us to develop a model so that we can scale it up or scale it down. If we have the jaws and the teeth that fossilize, which they do very easily, we can also compare that with extant animals and find out how big the animal was in history. So if you find something like megalodon tooth or megalodon jaws, that will then allow us to determine how long was the animal. So this group includes some of the largest and most successful vertebrate predators, sharks. When you get in the ocean, what do you worry about the most? Other than drowning, hopefully. You worry about sharks. Now, Rayfend fish, actinotergy, are the major lineages of the osteichthyes. Osteo. That means they have a bony skeleton as compared to the chondrichthyans, which have cartilage skeletons. So this clade of gnathostomes involves most of the vertebrates in the ocean. Nearly all of the osteichthyans have a bony endoskeleton and lung or lung derivatives. That does include the swim bladder. So most familiar aquatic osteichthyans are the Rayfend fish. And you see their fins here kind of look like rays of sun coming out. They have a radius, an ulna, and they've got carpal bones, just like you do. They have a scapula as well, shoulder blade. The lobed fin fish are the sarcopterygi. So these are the other major lineages of the osteichthyans. So again, they have a bony endoskeleton, but rather than having just these rays that are coming out, we start seeing more defined bones. So the bones are these rod-shaped bones that extend out from the body. It provides a very thick layer of muscles for their pectoral and pelvic fins. That means that they're going to allow for a better range of locomotion. 3 lineages survive today: the coelacanth, the lungfish, and the tetrapods. The tetrapods are the terrestrial vertebrates. Those have limbs and digits. They have a radius and ulna, the carpal bones, and fingers. So a comparison between the Rayfend fish, actinotergy, and the sarcopterygi. We still have the lobed fin fish, like the coelacanth, around, so we can compare this directly. The radius, the ulna, and the carpal bones, as well as the fingers, if you will. These fish don't have fingers. I get that part. There's no problem with that at all. But you can see that the radius, ulna, and the carpal bones are all the same types of bones that we find in our land vertebrates. So that tells you that the land vertebrates evolved from the lobed fin fish rather than the Rayfend fish. Well, how did they do that? That very small lobe right there is what allowed those animals to kind of push themselves up against gravity, allow them to pull themselves from one pool to another as the waters would recede or something like that inside, a marsh or a drying area, an area where the fish got trapped or something like that. They would then be able to move from one to the other. So you have a fish that can climb out of water into another pool of water. Those that were more efficient at it, or those that could then, like a mud skipper, they can survive outside of water. If they can survive outside of water and they have structures to move on land, those that do it more efficiently will survive on land better. And eventually you get to a point where you have just land-born animals. So we see these effects of the bilaterian radiations 1 and 2 that I've just mentioned. Large marine eukaryotes, very slow-moving and soft-bodied prior to the radiation, but afterwards we start seeing the vertebrates, the gnathostomes, are becoming the dominant marine predators for over 400 million years, very well-defined predator-prey relationships, and so on. Now, several animal groups have features facilitating their colonization of land. This is where we start seeing animals crawl out, just as I mentioned. So these profound changes start to develop our terrestrial communities. Reptiles, amphibians, arthropods, and mammals. Arthropods came first, long before everything else did. Amphibians then gave rise to the reptiles, mammals, and so on. Now, it was a divergence. It was not one transitioning into the other. The idea of, well, why are there still amphibians? We didn't come from the extant amphibians that we have. We came from a common ancestor with them. Reptiles would then give rise to reptilian-like mammals. Those would then give rise to mammals, and so on. So understand there is a lot of gray area in those transitions and the development of those characteristics. The populations evolve. So early land animals, we start seeing these advantages of being out of the water where we don't have predators, but we also see some challenges that are being, overcome, if you will. So the atmosphere had high oxygen concentration, which means lots of energy can be produced if we do it right. There are new food sources and fewer competitors now that land plants have developed and fungus. Arthropods are out there. Problem, water is much more scarce, which means you'll dry out if you don't develop some way of maintaining the moisture. Our cells have to have water. Temperatures fluctuate with greater extremes on the land than it does in the water. And there's no support against gravity. Members of animal groups made the transition to terrestrial life, the arthropods. So they came about 450 million years ago. Some arthropods developed winged and then powered flight. So they can go in the water, they can go on land, and they can live in the air. Vertebrates colonized the land about 365 million years ago. So for about 100 million years, arthropods were the only animals out there on the land. But once vertebrates were able to colonize the land, they kind of took over. So evolutionary changes that accompanied the transition to terrestrial life are much less extensive in animals than we find in plants. Plants derived and developed a great deal of adaptation that were not seen in their ancestors. Animals didn't have to overcome all of that. We'll pause, look at the chart, find out where things were derived or where it came from. If it was derived, where did that come from? You don't have to memorize this chart. Just know that it's there for you. The terrestrial lineages have arisen in several different arthropod groups, including millipedes, spiders or arachnids, crabs, and insects. Now, not all of those are just bugs. So the appendages of some living arthropods are modified for walking. Some are modified for feeding. Some are for sensory reception. You see the little antennae? They're used for reproduction and for defense. So appendages from which they were derived, those structures are jointed and paired. Arthropod. The foot has joints or jointed feet. For example, we have our sensory reception. Those are antennae. We have these little chelipeds here. Those are allowed for defense and for interaction with the world. We have some mouth parts. They kind of look like they used to be walking legs that are now used for feeding. We have walking legs, 8 of those. And then we've got some other portions. We have our genital region on the other side, and we've got our swimming appendages. Lobsters can swim. They swim in reverse. Those are just examples. So the body of an arthropod is completely covered by something that's called a cuticle. That's an exoskeleton made of layers of waterproofing protein and chitin. The exoskeleton gives structural support, allows for muscle attachment, and allows for protection from physical harm as well as from desiccation, which means drying out. It protects them from drying out. It protects them from predation. It reinforces their body. Unfortunately, it does also become a problem with gas exchange. You can only get so big. So a variety of organs have now been specialized for gas exchange. For example, insects. If you've ever watched something like a grasshopper, its abdomen will kind of compress and then expand and compress and then expand. That allows for gas exchange, oxygen in, CO2 out. Now, insects live in almost every terrestrial habitat and in fresh water, so they can live in marine, land, fresh water, and they can survive in the air as well for those that can fly. The oldest insect fossils date to about 416 million years ago. They've been around a really long time, and they're very successful at what they do. They outnumber us exponentially. So examples such as the lepidopterans, those are our butterflies. Hymenopterans, those are our bees, our wasps. And then we've got the hemipterans, those are our true bugs, and shield bugs, stink bugs. So insects diverged several times following the evolution of flight You see the adaptation of feeding on gymnosperms. Those are our things like conifers, and the expansion of the angiosperms. The angiosperms are our flowering plants. So you start seeing the animals adapt to the plants. Insect and plant diversity start to decline during the Cretaceous extinction, but both groups have since rebounded. Evolution of flight is one of the greatest successes ever. So animals that can fly can escape predators. They can find food easily. They can disperse to new habitats much quicker than they can if they can only crawl or walk. So because the wings are extension of the cuticle, they didn't lose a walking leg. They didn't give up one of their appendages. They developed a new one. We call that analogous evolution. That's where you get a different part using for the same function. For an example, birds, which arose from the dinosaurs, gave up one of their appendages. They gave up their front arms for flight. Now, an insect wing developed from the cuticle. They did not give up a walking leg, but they still fly. So it's a different part used for the same function. So we call that analogous evolution. Homologous means you use the same thing for a different function. Homologous evolution means that you're going to use something like the front arm for grasping, or you're going to use the front arm for swimming, like a flipper for a whale, versus something like a human hand. That's homologous evolution. So a key event in the colonization of land occurred when fins of the lobed fin fish developed into the limbs that we find in tetrapods. Tiktaalik, that's the Fisherpod. It shows an animal with both fish and tetrapod characteristics. It had fins, gills, lungs, and scales. The ribs used to breathe air and support its body against gravity. It had a neck and shoulders, which fish don't have. And fins, those are for the bone patterns of tetrapod limbs. And they looked a bit like this. So eyes on top of the skull so they could see out of the water, a flat skull like we would see with something like a crocodilian. It had a neck so it could move its head left and right, up and down. It had shoulder bones. It couldn't really support itself against gravity that well, but it did. The scales were still there, just like we would see in a fish. And then it also shows a humerus, an ulna, a radius, and the carpal bones. So a prime example of an organism that is showing the two different sides. Where did it come from and where is it going? So Tiktaalik could likely prop itself up on its fins while in the water, but it could not efficiently walk on land. It predates the oldest tetrapod. So the traits were then ancestral to the tetrapod lineage. That does not mean we evolved from it. We evolved from a common ancestor with it. Fins then become progressively more limb-like over time. Give it a long time, you start to develop these animals that can then get out of the water, crawl across the land from one pool to the next. So in Valentia Island in Ireland, about 380 million years ago, we see the oldest fossil tracks on land. The animal had a blade-like tail, so it could still swim. You could see that flopping back and forth. Here's one area where it flopped one direction, and here's one area here where it flopped the other. So the tail would go back and forth. But you can see the animal was walking like we would see in modern day lizards. It was not a lizard. They didn't evolve yet. So we start off very simply with lungfish. They can breathe air. We've got some others. That is a Fisherpod. It starts to develop these limbs so they can walk on, kind of muddy areas and marshes and so on. Later, we have Acanthostega. Later, we will develop the amphibians, and the rest becomes the amnioles. So amphibians are represented by about 6,100 species. So salamanders, frogs, and caecilians. The amphibians have gill-breathing aquatic larvae. So that means they have gills and they live in the water. They will then undergo a metamorphosis into a lung-breathing adult. Their skin can also help with that. They have a pulmocutaneous circulatory system. Pulmo meaning lung and cutaneous meaning skin. They must stay moist. They have to stay wet. So amphibians include those without an aquatic larval stage. Those are going to be restricted to damp habitats. They can't dry out. They don't have the ability to prevent desiccation. They will dry out. Their skin must stay moist to facilitate the gas exchange. So here's a salamander. It looks a bit like a lizard, correct? It's not a lizard, it's a salamander. Completely different organisms. That's convergent evolution. Frogs and toads, they're Anirians, they don't have tails. Okay, as adults, they do when they're polywogs or when they're tadpoles. And then caecilians don't have legs. They're burrowing animals. They like to be near the edges of the streams, but they're burrowing animals, but they must stay moist. Salamanders and caecilians don't have to go through that metamorphosis outside of the egg. Some of them go through that metamorphosis inside the egg. So amphibian populations are rapidly declining, and that's a problem. One, the chytrid fungus, Chytridiomycosis, habitat loss. We keep using their land. They don't have any place to go. They have to be near the water. Climate change and pollution. If you want to see how healthy an ecosystem is, look for the amphibians. Second, look for the rotifers. So at least 9 amphibian species have gone extinct in the last 40 years. More than 100 others have been lost at that time as well. Now, amniotes brought the water with them. They don't have to leave their young in the water. They started to develop the egg in some way, or they developed the young inside their body. So amniotes are tetrapods that now become the reptiles, including the birds and mammals. So amniotes are named because of the character clade known as the amniotic egg. It contains 4 membranes that protect the embryo. It has an amnion, a chorion, a yolk sac, and an allantois. They look like this. So we have this egg with the albumin or ovoalbumin around the outside. We've got a yolk that provides nutrients to developing or growing embryo. We surround that with the amniotic cavity, amniotic fluid. We brought the water with us. Once we left the water, we brought the water with us. And then you cover that with a shell on the outside. That can be leathery or it can be more mineralized. So the amniotic egg for most reptiles and mammals have a shell. The shell slows dehydration. There's a small problem with that, is that you still have to diffuse oxygen into the egg. Now, most mammals have lost the eggshell. The embryo develops inside the amnion, inside the mother's body. Temporarily, like those that we find in the Metazones, like the, Metatherians, like the marsupials, or a little bit more long-term with those that are the uterine animals, or the eutherians. Another important adaptation for amniotes is the use of a rib cage to ventilate the lungs or breathing. Efficient ventilation for the rib cage allowed for less permeable skin, so we don't have to stay near the water, and better water conservation. We don't lose as much. We don't dry out as quickly. Now, living amphibians and amniotes split between the 2 lineages. Early amniotes lived in very warm, moist habitats. They adapted and expanded from there. Amniotes include 2 large terrestrial clades, the reptiles and the mammals. So reptiles started to evolve about 310 million years ago. Living members of the reptile clade are the tuatarans. Some of my favorite are tuataras, the snakes and lizards, the squamates, the turtles, chelonians, the crocodilians, and the birds. Birds originated from a common ancestor with the dinosaurs. Reptiles comprise 2 large clades, the tuatarans and the squamates. The others are smaller in size, the turtles, crocodiles, and the birds. Now, the ornithischians, the pterosaurs, and the saurischians are all extinct. Pterosaurs, or pterodactyl, those were not dinosaurs. They branched off earlier than that. They were just flying reptiles. But the ornithischians and the saurischians gave rise to the dinosaurs. Those are the types of dinosaurs. The saurischians, those gave rise to the birds, and the birds survived the extinction. So did the tuatarans and squamates, the snakes and the lizards. So reptiles have some shared derived characteristics, such as the scales that waterproofed the skin, which means they didn't dry out. They have internal fertilization and lay a shelled egg. Most are ectothermic, which means they absorb heat from the outside, which means they're very efficient. Unfortunately, they have to work at regulating their body temperature so they can stay, active. Birds are a very diverse group of flying reptiles. You can still see their scales and their feet if you ever get a chance to look at a bird's foot. They evolved about 160 million years ago. They lack a urinary bladder. They don't hold on to the water. Females have one ovary. That means they decrease their weight. They have smaller gonads, except during breeding season, that increases, and they don't have teeth. Now, early transition animals actually had teeth. There were toothed birds at one point, but they weren't true birds. They had their own classification. Notice how those small changes over time add up, and that when we have this change in allele frequency over time, we lose characteristics or we develop new characteristics. It takes a while, and it's not one organism that does so. It's a population that does so. That's how evolution works, is through the population. Now, other adaptations for flight include the wings and the feathers, of course, very large pectoral muscles, like the breast of a chicken, a keel on the sternum, which means you're going to anchor those very large pectoral muscles, efficient respiration and circulatory systems. Birds breathe in and breathe out, just like we do. But each time they breathe in, they also fill their air sacs. And when they breathe out, those air sacs send the air again across the lungs. So they get oxygen breathing in and breathing out. We also see the development of a four-chambered heart. We see color vision and acute eyesight develop, as well as visual and motor areas of the brain. Different types of structures start showing up. The scales on reptiles, the feathers on birds, and the hair on mammals are all the same types of proteins. They just modified them, just like we saw with modification of the Hox genes earlier. The trochata retains its claws. We can see that here. See it right there? There's a claw. There's the finger right there. There are birds that still have claws. The Hoatzins, another one, but they only maintain theirs as a chick. You can see the claw right there and right there. Brilliant birds, absolutely brilliant. The fact that they still maintain some of their reptilian characteristics. Now, mammals and other extant lineages of amniotes also have some derived traits, such as mammary glands that produce milk, the development of hair, subdermal adipose tissue, known as a fat layer under the skin, that also later gives rise in whales to blubber, the kidney, which conserves water, high metabolic rate due to endothermy, we generate our own heat, a relatively large brain, and differentiated teeth. About half of all mammal species burrow. Mammals evolved from synapsids or synapsids. So we have one cheekbone as compared to those that are like mammals, or that are reptiles, like the dinosaurs. They had two. So they had diapsid animals as well. That's how we know whether or not a fossil is a dinosaur or a mammal. So about 140 million years ago, the three lineages of mammals start to emerged with Prototherians, the monotremes. Those are egg-laying mammals, such as the platypus and the echidna. The marsupials, known as the Metatherians, those are mammals with a pouch. And the eutherians, those are placental mammals. So here's an echidna there on the top left. Other marsupials, like the Metatherian kangaroo, and the eutherians, such as this orangutan. So primates are a group of eutherians. They involve the lemurs, the tarsiers, the monkeys, and the apes. Humans are a member of the ape group. We are the Eastern African great ape, if you will. We start seeing all of these developments show up, and then later on we'll develop into humans and the other extinct Homo species, like Homo neanderthalis. So derived traits from primates are adaptations for lives in trees, like grasping hands and feet, separate movable thumb or opposable thumbs. Monkeys and apes have opposable thumbs. Large brains and short jaws. Our faces are getting shorter. And then forward-looking eyes for great depth perception to see where one branches from the other. Non-human apes are only found in tropical regions, such as Africa and Asia. Apes have larger brains in proportion to their body and more flexible behavior. That is the important characteristic right there. More flexible behavior, which means they adapt very well. Humans are primates nested inside that group, informally known as apes. We've got a number of characteristics that distinguish humans from other apes, such as upright posture and bipedal locomotion. We started walking upright first, then we developed these very large brains capable of language, symbolic thought that can be sent from one generation to the next, or laterally from one person to the next, artistic expression. We start drawing and we start using tools. Now, chimpanzees are the closest living relatives to humans with a genome that's about 99% identical to our own. There's still some pretty major differences, though. So the lineage that leads to humans diverged from chimpanzees about 6.5 million years ago or so. So humans and their extinct human ancestors comprise the hominins. Evolution of bipedalism preceded the evolution of the increased size of our brain. So we started walking upright first so we could see over those roads, grasses. We could see really far away so we can find out where exactly was the predator. We could run or at least get out of the way of the predator if necessary. So brain size, body size, and tools start to increase over that time as well. But once we become bipedal, the trees don't really support us as well anymore. So we, rather than stay in the trees, start walking across the savanna. Here's Artie with his little opposable toe, Artipithecus. Ramidus means branch, and Artipithecus, Artie, that's we'll just leave it at that. Just understand that with Artipithecus, they still had a toe that stuck out like a thumb. So that was one of our earliest ancestors. Okay. Now, leading up to Homo sapien, the oldest Homo sapien fossils are from 2 specimens in Ethiopia. They date from around 195,000 years ago to about 160,000 years ago. All living humans have ancestors that originated with Homo sapiens in Africa. Humans dispersed out of Africa into Asia, and then into Europe, Australia, and then eventually into the New World. The shared morphological characteristics and the analysis of the DNA extracted from the fossil human jawbone indicate that humans interbred with Neanderthals. We know that we share somewhere between 4 and 6% of our DNA with Neanderthals. That excludes anybody who is of the San or the Yoruba people in Africa. They don't have that shared characteristic. They don't have any Neanderthal DNA. So animals have transitioned and adapted their environment so much so that they actually transform the ecosystem. For example, beavers create these dams, which can then create a flooded area. They're changing the entire world at that point. Okay. So the ocean of the early Earth was likely very different than it is now, specifically with the dissolved amounts of iron that was in it. So it was very murky or so. It was kind of a greenish color as compared to the nice pale blue that we have now. The rise of suspension feeding animals then show a decline in that cyanobacteria. We start clearing all of that out. The algae gets cleared out, if you will, or at least the cyanobacteria. The algae then becomes the dominant producers. So they're going to start producing all of this food chain or food web, as well as oxygen and so on. They establish the feeding relationships. They anchor the entire planet's food system. For terrestrial ecosystems, we start seeing animals move on land. Herbivores then improve the growth of plants at low population sizes because of nutrient-rich waste products. They move the nutrients around. At high population sizes, herbivores can defoliate very large tracts of land. They can actually overeat the plants. Predators can also transform landscapes through the regulation of herbivore populations. So if the herbivores are eating too many plants, throw in a predator and they'll take care of that problem. The origin of mobile heterotrophic animals, which means they're going to get up, move around, and eat other things, with those complete digestive tracts, drove some of those species to extinction. There's that arms race again. Once you start moving around and becoming efficient at collecting other animals to eat, that starts that arms race again, or it maintains it, it reinforces it. So 2 species that interact exert very strong reciprocal selective pressures. For example, a flower formed can then influence how an insect is formed, and how the insect is formed will also influence how that flower is formed. For example, the proboscis here. You see on this moth, the proboscis is really long. Well, it's going to reach way down here to get the nectar. And by doing so, this is the only animal that can pollinate this flower. That also means that this flower is the only flower that this moth will be able to get food from. So they work back and forth with each other. So those reciprocal selection pressures occur when you see a new species being added. Okay? So the origin of new groups of animals provide new food sources for parasites, new radiations of parasite groups, and then the adaptations to those as well. So humans change the environment. They start altering the selective pressures in lots of these different species. Human beings are the selective pressure on most of the organisms on this planet. Humans targeting very large amounts of fish, I call that overexploitation, has led to a reduction in the age and the size in which the individuals can reach sexual maturity. For example, way back, they were able to catch huge fish, very large amounts of them. These were old fish. Then we got to a point where they were slightly smaller. They were reaching sexual maturity way before they reached this size. And now we get to a point where there are very few left at all. And these are prize-winning fish. So rapid declines over the last 400 years indicate that human activities are the driving force for our sixth mass extinction, known as the Anthropocene extinction. Mollusks have suffered the greatest impact. So 3 major threats: habitat loss, pollution, and then competition, also known as predation, as well as overharvesting being the same thing. When we pull out those species, we are competing with them for their own life. So I combined competition and predation and overharvesting. I combine those 3. Pollution and habitat loss are the other 2. Okay? So take care of your planet and all who inhabit it.